There are different ways in which cells regulate gene expression at the level of transcription. In order for transcription to actually occur, the genes need to be first accessible to the RNA polymerase. So cells can regulate gene expression by controlling access to genes. One way to control access to genes is simply by changing the structure of chromatin, and that's accomplished by modifying the amino acids on the tails of the histones. There are two major types of modifications that can be carried out. These are acetylation and methylation. As the name suggests, acetylation is the addition of acetyl groups. Now, the effect of this is to destabilize the nucleosome. And so what this simply means is that the addition of the acetyl group loosens the chromatin. So it makes this chromatin more accessible to RNA polymerase and transcription factors. So histone acetyltransferases, which are the enzymes that add the acetyl groups, would make chromatin more euchromatic. On the other hand, the histone deacetylases, which remove the acetyl groups, would have the opposite effect. They would stabilize the nucleosomes, and as a result, the chromatin would be more heterochromatic. When we look at histone methylation, which is the addition of methyl groups, the situation is a little more complex. And I want to be very clear here. What I'm talking about is histone methylation, not DNA methylation, at least not yet. So this is what I'm about to say refers to histone methylation. Now, histone methylation usually represses transcription, but it can also be associated with transcriptional activation. So that means that histone methyltransferases, which add methyl groups, and histone demethylases, which remove methyl groups, can be associated with either transcriptional activation or repression. So the addition of a methyl group can either make that chromatin more euchromatic or more heterochromatic. Whether histone methylation stabilizes or destabilizes nucleosomes is going to depend on where the methyl group is added. So depending on the amino acid that is methylated, the effect of the methylation is going to be different. For example, the addition of a methyl group to lysine number 4 of histone 3 promotes the destabilization of the nucleosome so that the chromatin becomes more loose or euchromatic. And so this type of methylation is associated with transcriptional activation. Methylation of lysine 9 on histone 3 has the opposite effect. It promotes the stabilization of the nucleosomes so that the chromatin becomes more tight or heterochromatic. So this type of methylation is associated with transcriptional repression. So once again, what this means is that the activity of the histone methyltransferases and the histone demethylases could either be associated with transcriptional activation or repression because the effect of the addition of the methyl group depends on the specific amino acid to which the methyl group is added. A very interesting thing about histone methylation is that cells are actually able to retain their memory of methylation through cell division. So what I mean is that a cell can maintain the same pattern of histone methylation even after going through mitosis. And there are two families of proteins that are involved in that, trithorax and polycomb, and you're not going to have to remember the name. The important thing to remember here is how this happens. So the trithorax proteins, they're associated with the maintenance or the inheritance of transcriptional activation. So they act to make the chromatin more euchromatic. So what are they going to do? They're going to promote the addition of methyl groups that are associated with transcriptional activation, and they're going to help to maintain those changes. On the other hand, the polycomb proteins are associated with the inheritance of transcriptional repression. So they're going to act to make the chromatin more heterochromatic and to maintain that state through cell division. So they'll promote the tightening of the nucleosomes by the addition of methyl groups at locations in the histones that are associated with transcriptional repression. Whereas histone methylation can either activate or repress transcription depending on where the methyl group is added on the histone, DNA methylation has a different effect. If we're talking about promoters and enhancers, the addition of methyl groups to DNA is associated with transcriptional repression. DNA methylation is not seen in every gene, but it is significant because it occurs in approximately 5% of mammalian genes. Now, an important thing to note is that the methyl groups on DNA can only be added to cytosine. So cytosine is the only base in the DNA that can be methylated. And this is important, you'll see in a moment, because it allows for the inheritance of the DNA methylation pattern. Not only that, the cytosines that can be methylated are only those that are followed by guanines. So methylation can only happen on a cytosine if there is a guanine that comes after that cytosine. Otherwise, the methyl group will not be added to that cytosine. The patterns of DNA methylation can change over time, just like the patterns of histone methylation. For example, in cells that produce hemoglobin, the promoter for the embryonic hemoglobin gene is unmethylated during the first 6 weeks of development. So embryonic hemoglobin is produced at this particular stage. At 12 weeks, the pattern of DNA methylation changes so that the promoter for the embryonic hemoglobin is now methylated, and thus the gene is inactive, while the promoter for fetal hemoglobin is now unmethylated, and thus fetal hemoglobin is being produced instead. So what does DNA methylation actually do? How does the addition of a methyl group to a promoter or enhancer repress transcription? Well, as you can see, DNA methylation acts by blocking the binding of transcription factors. Now, since transcription factor binding to promoters and to enhancers is needed for transcription to occur by recruiting RNA polymerase, for example, and by recruiting the transcription initiation complex, the lack of a transcription factor at the enhancer will prevent the gene from being activated, so the gene will not be expressed. Another way DNA methylation acts to repress transcription is by helping to recruit histone modifying enzymes. More specifically, the modifications on the DNA, as you can see here, will lead to the recruitment of histone methyltransferases and histone deacetylases. So this is going to have the effect of making the chromatin more heterochromatic. So the methylation on the DNA is not only going to prevent the binding of transcription factors, but it will also recruit these proteins that are going to lead to the stabilization of the nucleosomes. And in turn, that's going to repress transcription because it's going to make it more difficult for other transcription factors and for RNA polymerase to bind to that region. Recall that DNA methylation can only happen in cytosines that are followed by guanines. This restriction is important for maintaining the memory of DNA methylation through cell division. So when the DNA is replicated, the newly synthesized strand needs to be methylated in accordance to the pattern of methylation of the template strand. Now, the DNA methyltransferase that establishes the pattern of methylation on the new strand is able to recognize methyl cytosine, so it can tell that this is a methyl cytosine on one strand, and then add the methyl group to the cytosine on the newly synthesized strand. So this is why the methyl groups are added only to C's that are located next to G, so that the methyl group will be added to the new strand at the C that is complementary to the G that follows the methylated cytosine.